Hello friends, welcome back to my YouTube channel Nesting Padasale. So currently we are revising for GATE exam 2023. So in case if you are applying for GATE examination under biotechnology paper or life sciences paper, then this video will be really helpful. So in this today video, we are going to study about the common reagent or common enzyme that are used for protein cleavage. So you might be wondering why these reagents or why a protein need to cleave. So generally these reagents are employed for protein sequencing studies, which are generally used for determining the primary structure of amino acid. Okay, so these are the list of reagent along with the biological sources. So first we'll be studying the reagent and the biological sources, and next we'll be see at which point of amino acid and what is the cleavage point of these reagent. Okay. So first thing is trypsin, which is isolated from bovine pancreas. Next is submaxillaris protease, which is isolated from mouse submaxillary gland. Next is chymotrypsin, which is isolated from bovine pancreas. Next is Staphylococcus aureus V8 protease, which is isolated from bacteria Staphylococcus aureus. Next is asparagine N protease. Okay, so it is isolated from a bacterium Pseudomonas faragi. Next is pepsin, which is isolated from frozen pig stomach of pig. Next is endoproteinase lysine C, which is isolated from a bacterium called Lysobacter enzymogens. Okay. Next is cyanogen bromide. This is a chemical. So first we'll be seeing the cleavage point. So before that you should know the amino acid. So all peptide will be having a 2N. One N will be consist of free amino group, that is N terminal N. The other N will be consist of free carboxylyl group or COOH group. First, we will be seeing trypsin. So all most commonly these reagent will be cutting an amino acid at a carboxyl terminal N only, except this asparagine N protease and pepsin, which will be cleaving at a N terminal side chain. Okay, so here we can able to see first is trypsin. The cleavage points of trypsin is that it will be cleaved at a carboxyl terminal end of lysine amino acid and arginine amino acid. So here you can able to see it is cleaving the right side or C terminal end of trypsin. With the arrow mark, we can able to see the C terminal end. This is a N terminal end and this is a C terminal end. Next is this particular submaxillaris protease, which cleave at arginine in the C terminal side, and next is chymotrypsin. Chymotrypsin again cleaves at C-terminal end, but it will be cleaving after phenylalanine, C-terminal end of phenylalanine, tryptophan and tyrosine amino acid. Next is Staphylococcus aureus V8 protease, which will be cleaving the C-terminal end of asparagine and glutamine. Next is with regarding to this asparagine N protease, which is cleaving this. So for Staphylococcus aureus and asparagine N protease, both will be cleaving after this asparagine and glutamine only. But Staphylococcus aureus V8 protease will be cleaving at a C terminal end, whereas this asparagine N protease will be cleaving after amino terminal N. Next is pepsin, which will be cleaving at a N terminal or amino terminal end of leucine, phenylalanine, tryptophan and tyrosine. Okay, so here you can be able to see easily. Next is cyanogen bromide, which will be cleaving after at a methionine. So here you can able to see in this amino acid sequence methionine is there, so it will be cleaving at a C terminal side chain of methionine. Okay, same elastase is again an enzyme which is used for cleaving of polypeptide chain. It will be cleaving after glycine amino acid, that is in C terminal side. Next is subtilisine, which is will be cleaving at the C terminal end of phenylalanine. Next is carboxypeptidase, which will be cleaving the C terminal side chain of aspartic acid. Next is conjugated protein. So protein with amino acid alone, amino acid as a building block is called a simple protein. Some protein will be consist of additional units, so that's why these protein are named as conjugated protein. The example of conjugated protein include lipoprotein, glycoprotein, phosphoprotein, hemoprotein, flavoprotein and metalloprotein. Okay, so we are going to see prosthetic group, which is nothing but additional group which is present along with the protein. So lipoprotein, the prosthetic group is lipid, glycoprotein it is carbohydrate, phosphoprotein it is phosphate group, hemoprotein it is heme or iron porphyrin ring, flavoprotein it is flavin nucleotide, metalloprotein consists of 5 different phosphatic group, that is iron, zinc, calcium, molybdenum and copper. So we will also discuss the example. Example of lipoprotein is beta-1 lipoprotein of blood. Example of glycoprotein, it is immunoglobulin G. And phosphoprotein, example is casein of your milk. And next chemoprotein, the example is hemoglobin. And next flavoprotein, it is a example of flavoprotein is succinate dehydrogenase. So succinate dehydrogenase is a conjugated protein which is present in the inner membrane of mitochondria. This succine require a flavin, that is FMN or FAD as a cofactor or coenzyme. Okay, so this succinate dehydrogenase is involved oxidation of your succinate to fumarate. Next, with regarding to metalloprotein, ferritin is an iron-containing metalloprotein. Alcohol dehydrogenase consists of this enzyme has zinc as a prosthetic group, calmodulin, which is an extracellular matrix protein, which need calcium as a prosthetic group for its effective function. Next is dihydrogenase, which require EMBO, that is molybdenum as a cofactor. Okay. Next, plastocyanin, which is present in plant, it requires copper as the prosthetic group. Okay. So ferritin, alcohol dehydrogenase, calmodulin, dihydrogenase, plastocyanin, all are belonging to metalloprotein and all are conjugated protein. Next, we'll be seeing a comparison